Hello, Bio 107 students. Welcome to Lab 10. This lab is on DNA and genetics, and I just wanted to show you some of the materials we have for the lab. Uh, the first thing that you'll do is complete the virtual lab and related assignments. You're watching the introductory video. The virtual lab has some background material, background information that goes along with it. That's on the McGraw-Hill website and Connect. Then you'll read the case study and analyze the results. This is located down here on Moodle. So this is a case study about breast cancer. And there are assignment questions located in the case study. So as you read through, you might notice some questions. It's good to answer those as you go, and then you can go back and finalize them for your submission for your assignment. There's also a pedigree chart in there, and a pedigree chart, as you're going to see, is like a family tree for a biological trait. So we're looking at heredity here. The next thing that you'll do, this is hands-on, a fruit DNA extraction. It's a lot of fun. You can use any kind of fruit. Strawberries and bananas tend to work best. And you're going to make an extraction buffer using salt and dish detergent. So something that you use to wash dishes like Dawn or Joy, Palmolive, those types of dish detergent, not what you put in a dishwashing machine. That's actually different. So you want the detergent type thing right next to the sink. And you'll be mashing up fruit and then using alcohol to separate the solution. And you can use vodka, you can use other spirits or rubbing alcohol, whatever you have on hand. And like with previous hands-on labs, you're welcome to substitute materials, and feel free to email me if you have a question about a substitution. I want to keep it as simple as possible, but still have the results we're looking for. And don't forget to take the quiz. There are some optional PowerPoint slides that go along with this as well for some images and more information. So take care and good luck.